Good morning, good afternoon, and good evening. Just want to welcome all of you to our next session on equity and inclusion. As part of the Human Cell Atlas, we are making strong efforts to ensure that all the data that we generate that we can have a complete Human Cell Atlas. We have a very exciting list of speakers ranging from various countries. I just want to highlight a statistic that Alistair had mentioned yesterday, that we have new participants, especially from new countries in Asia, joining this HCA Asia meeting compared to last year. So this is a great indication that our message to be more inclusive is being sent out, and that more members and more countries from Asia are participating in this meeting, which means that there's more opportunities to be more equitable and collaborative in ensuring that we can complete the Human Cell Atlas altogether. So I think we're going to have a very exciting talks as well as discussions towards the end of the session. So please feel free to ask questions, and we'll have a discussion towards the end. So let's get started with our very first speaker, Dr. Partha Majumder. You may have heard him in many HCA meetings. He is one of the committee members, as well as one of the key leading members of the equity working group of the Human Cell Atlas. He is a professor and founder of National Institute of Biomedical Genomics, and he's also the president of Indian Academy of Science. So with further ado, let's invite Dr. Partha Majumder, and we look forward to your presentation. So please go ahead.
Thanks, Jay. Thank you very much. Good morning, good evening, good afternoon, everyone. It's really my pleasure to present the equity and ethical imperatives. Let me share my screen. All right, so I'm going to talk about the ethical equity imperatives primarily, and as Jay mentioned, I'm a co-chair of the HCA Equity Working Group, and this presentation is essentially on behalf of a large number of people and committees. I'm presenting this on behalf of Musa Malanga and Alex Shalek, who are the co-chairs of the Equity Working Group. And this is based on some write-ups that we prepared, and the write-up was initiated by the Equity Working Group, but has now been vetted by the organizing committee, primarily Aviv and Sarah, and other members of the HCA organizing committee. It's also been vetted by Partha, who is the chair of the ethics working group, and members of the ethics working group. As all of us know, that the HCA is intending to reconstruct the human body, reference human body, or reference human bodies, cell by cell, organ by organ, for every organ of the human body. And in trying to do so, it has a mission that demands inclusive representation. And this inclusive representation is essentially to build a reference that is representative of different genders, ages, ethnicity, environments, and socioeconomic status. The reason why this needs to be done in an inclusive manner is because HCA's success depends on it, because this reference will actually reference our ability to understand disease as well. So equity is a point of emphasis of HCA, has been a point of emphasis of HCA from its inception in 2016. What we intend to do is that we want to obtain a view from a diverse set of people, and obtaining such a view will help us better understand human biology, will also help us contribute, understand the contributions of our environments and genetics to the ways in which we keep ourselves well. For this, global participation is critical. A necessary condition is the ability to generate and analyze data locally. So that's what the equity is pushing for. We do not want to do helicopter science and just collect samples from various parts of the world and disengage scientists from scientists locally. Instead, we wish to engage scientists locally and get the data to the extent possible, generate the data locally, and derive global benefits from being local and as also being inclusive. Our mission critical is make the HCA socially and scientifically distinctive, and we really do believe that this is the time when it needs to become socially and scientifically distinctive, because sometimes the history does not show this. And we want to do this through commitment to global engagement. We do want to create an active outreach and education program, and I'll present to you some of the things that we have done and we are in the process of doing. Although the pandemic came in the way, otherwise there would have been much better progress. So we do want to build local capability, and that's something that's on the top of our minds, and we are working towards that. And we want to forge and foster local collaborative partnership, without which we don't think that HCA will succeed. The point is that we can all benefit from diverse community perspectives and shared approaches to facing complex challenges in human biology. Especially the complex challenges arise because our ancestries are different, because we live in different kinds of environments, and therefore to be able to understand human biology, we need to collect data from these diversities. We've learned some lessons as we went along. The first lesson that we have learned is that we don't gain trust without giving trust. So we need to trust people. We need to give trust to people, and thereby earn trust of people. For the HCA to be representative of the people, we believe that it must be erected by the people. It has to be done locally. It must be erected by the people. There must be inclusivity. And it must be for the people. We can't think of different kinds of projects that will not benefit humanity, that will not benefit humankind. So it has to be erected by the people. It has to be for the people. And another important thing that we have learned is to keep our ears open. And it's important to ask questions and listen carefully to the answers. And sometimes the information, approaches, and initiative that we believe are common knowledge, actually we have learned that they are often in fact not. Inequity and that lack of information, lack of keeping our ears and eyes open can actually lead to promotion of inequity. It can be driven by lack of knowledge and lack of infrastructure, resources, and capacity. And some of it can be real, some of it can also be perceived. So talking helps. Talking helps demote perception, wrong perception. We wish to identify, articulate, and track how equitable partnerships on multiple levels lead to long-term empowerment, mutual benefit, and equal participation. So without equitable partnership, these kinds of things will not happen, and HCA's mission will not be successful. Like I said, that we need to engage different communities and ethnicities, and we do not want to do helicopter science. We've opened up membership both to the HCA as also to different working groups of the HCA, and each working group and the HCA has certain value systems. And for somebody to become a member to either the working groups or to the HCA, one must commit an adherence to these values. That's very critical. Not everybody who does not subscribe to these values can become members. Reaching out to diverse communities with intent to scientifically empower and support. And as I said, that we just don't want to descend on a community or on a geographical region, collect samples and rush out of there not even go back to the community. So we do want to encourage, in order to be able to scientifically empower, we want to encourage local scientists to lead HCA projects. Local data generation and analysis. As you can see that local data generation, this decentralization is at the core of HCA. And to the extent possible, we are trying to do that. And of course, if the data are generated locally, the results are shared. Some data may not be It may not be possible to generate some data locally. Even so, wherever it's generated, we go back to the local communities and share the results and provide explanations as to what the implications of those results are. We want to make sure that such data are not used for any form of discrimination. And we want to underscore this primarily because history of human genetics is not without such blemishes. And so we do want to emphasize that we do not want these data to be discriminatory in any form or to discriminate among communities in any form. The HCA considers racial profiling or any form of discrimination to be against the spirit of the consortium. And to be against the purpose of data collection. The purpose of data collection is to be inclusive, is to be equitable, and the ethical conduct expected to be followed by consortium members. And we do believe that consortium members should take a pledge not to use the data for any form of racial discrimination or any form of discrimination whatsoever, genetic discrimination whatsoever. There are some success stories, and this is I'm only presenting the success stories of the equity working group. That was formed almost concomitantly with the formation of the organizing committee. It's now fully constituted with diverse international representation. It was initially the 3 of us, but we have now expanded. There are 15 members covering 6 continents, 14 countries, and it's not a completely balanced gender representation, but 10 males and 5 females. We have started meeting regularly to discuss initial efforts and how to build local networks. And the reason why we expanded essentially is to be able to enable us to build local networks. Latin American HCA has hosted their first HCA general meeting about 2 years ago. It's now involved multiple Latin American countries all the way to Mexico. Focus areas have been developed, and the focus areas are local problems, dealing with local problems. And of course, even though HCA pertains to the normal human body, the reference is for understanding human disease, and these diseases have also been chosen depending on what the local conditions are. So cancer, parasitic disease, for example, are discussed in the Latin American calls, with collaborations as far as Cuba, who are scientists of Cuba participating. The Sao Paulo Research Foundation has expressed an interest in supporting a 10-year central initiative. And the initiative or the proposal is to bring together academics and industry to catalyze single-cell biology with joint funding from the Sao Paulo Research Foundation, University of Sao Paulo, and external sources. There are now outreach presentations that have been done in India and in Saudi Arabia, and there's more coming. There have been some roadshows, in-person roadshows before the pandemic. There was a roadshow in Brazil, which was hugely successful. And this roadshow led to the formation of plans to do more roadshows. The roadshow that's at an advanced stage are in India, Vietnam, and Ghana. The plans for Vietnam are very advanced. Again, it would have taken place but for the pandemic. Early stage plans for Ghana are now underway. Ghana may serve as a hub for West Africa and HCA initiatives. So we have some local scientists there who are taking enormous interest, and they are coalescing, catalyzing the formation of networks, not only in West Africa, in a single country in West Africa, but across the region as well. Activities in India are also being planned, and we actually had a conference call last evening and to plan a workshop in India, which will probably also include some neighboring countries like Bangladesh and Pakistan. Virtual training resources and conferences have been planned, and there are some these are being explored, and some explorations have already been initiated. There are discussions with funding agencies happening because these things would require some level of funding, and these talks are ongoing. I think with CZI and with Wellcome Trust primarily, and also with USNIH. So there are discussions that are being held in order to obtain some funding so that we can promote equity, we can promote equitable participation in the HCA. There's also a resources tab that has been created on the HCA website, These contain curated online training materials and resources. It's a kind of, there are some portions of it that are kind of sketchy, and more meat needs to be added to this such that we have a greater library of educational content, and that has to be done by multiple people, and that will be done over a period of time. And we've sown the seeds for that, and we hope that the tree will grow, and with contributions from various parts of the world. The Human Cell Atlas has been very supportive of showing around the world how its science is relevant to COVID-19. As many of you may know, that there have been lots of publications from members of the HCA. The bioarchive and medRxiv are full of papers that are kind of in the process of being formally published, but and this has all been catalyzed, been pushed, energized by the Human Cell Atlas. So that's all I had to present, and I'm happy to take any questions if there's time at this stage.
Great.
Thank you very much.
Thank you very much. Thank you very much, Partha. And it's really, really thankful and grateful for what you do for the Human Cell Atlas. And some of the resource that you're introducing is very exciting. And if there are any questions at the moment, maybe I'll throw in one quick question before the next speaker. So you mentioned your effort is global, meaning not only in Asia, but of course in Africa and Latin America, and of course here in Asia. What are the common threads or common themes that you see across different regions? And what are also the regional specific features that you see that we could also learn from one another?
So Jay, one of the themes that we see, oh well, more than themes, one of the feeling that we have as we are talking to different countries in Asia is that the level of preparedness is highly variable. So different, some countries have already started to contribute, and some countries are not even prepared. Both, neither they have the capacity, nor they have the infrastructure, nor they have the knowledge. And you can't, gain knowledge without actually dirtying your hands in some ways. So right now, the essential feature that we are trying to do is to promote the knowledge of HCA and the knowledge of the methods by which to do single cell transcriptomics. And we hope that these efforts will infuse them, and they will apply to their funding agencies and be able to generate, or international funding agencies, and be able to generate some funds to create these infrastructure and get the resources and define problems for themselves in order to get started. And we are all there, especially those of us who have been in this business for some time in various countries in Asia and also globally, to help them get initiated and do well. So that's what it is. There is a huge amount of diversity in terms of preparedness. So we need to take that into account quite reasonably. Yeah.
Great. That makes a lot of sense. And I think your idea of virtual video resources, perhaps, and how we can share that globally, I think that would also be a very good approach to reach out to everyone. So thank you very much. And we will save additional questions towards the end of the discussion. So now I'll hand over my chair to my co-chair, Dr. Hyun-Suk Lee. Please.
Hi, everybody. I'm Hyun-Suk Lee from Seoul National University. Our next speaker is Jonah Ko. He's a science program officer, the lead in single-cell biology program in Chan Chocobo Sciences. Jonah?
Great, thank you very much. So thank you to the organizers. It's really a pleasure to be here. The HCA Asia meeting is one that I look forward to on an annual basis. So nice that it's a much shorter flight, although I do regret that we're not all together. So hopefully that can happen again very, very soon. So in the next couple of minutes, I'm going to give you a little bit of an overview of some of the work within the Chan-Zuckerberg Initiative, specifically focused on supporting the first draft of the Human Cell Atlas. And there's a lot to cover. And fortunately, there's a number of other speakers that will touch on different points. And so what I really want to try to emphasize are a few points that I think Partha has teed up really well, which is opportunities to get engaged, some examples of how groups have become engaged in CZI-funded efforts, and then also the resources and output that hopefully are relevant and helpful to all of you. One thing maybe to note, which I think is maybe a late-breaking bit of news, is some of those virtual meetings and outreach that Partha alluded to, I think are now supported by CZI, and so really, really excited to hopefully broaden that reach and offer more opportunities to the community. So would encourage everyone to get engaged and support those efforts. So let me start with just an overview of the single cell biology program at the Chan Zuckerberg Initiative. So we are fundamentally focused on supporting the generation, assembly, and use of references that clarify cellular mechanisms of disease. And the primary way that we've supported this is through support for the Human Cell Atlas over the last number of years. And there's 4 pillars that we look to support in this effort. So one is the generation of reference data, and I'll talk about that. And that, of course, includes many different groups and investigators focused and located within Asia. It involves the development and refinement of tools and technologies. And here I'm referring to tools and technologies to be both the reference atlas itself, but also the methods that are used to generate that data. And perhaps most importantly, and something that I'll end on, the tools and technologies and computational and analytical tools required to analyze and gain more insight from that data. We are, of course, interested in using these tools and technologies to clarify diseases, and in particular diseases that are most interesting and important to you and the communities that you live within. And of course, all of this is supported, and we're very focused on building collaborative, and I've underscored this, but international communities. So we are a philanthropic funder. We are not a government and national funder, meaning that we have the flexibility to support work around the world, and perhaps most importantly, support collaborations that are international in nature. And so it's a great opportunity to help provide glue to these various areas. And then finally, some technology that hopefully helps the interplay of all of these different areas is a tool that CZI is developing. It's a close collaboration and connected in intention with some work in the Human Cell Atlas data coordination platform that I think you will hear about next. And that tool is called Cell by Gene, and I'll touch on that towards the end of the talk as well. Okay, so let me start with an overview of our support to date. So thus far, we have had 5 open calls for applications. All of these RFAs are open to international applicants anywhere in the world, so absolutely include you. It has resulted in the generation of data from approximately 20 million cells. This number is, of course, growing very rapidly. And again, to kind of keep this thread of international collaboration, it is to date included labs in 26 different countries covering all of the inhabited continents. So currently no grantees on Antarctica. That may change too, you never know. So this is what the CZI single grantee networks look like over time. And the point that I'll highlight here is in the first pilot projects, they're international indeed, but relatively consolidated or located primarily in Europe and North America. And then over time, with collaborative computational tools, you start to see some groups in Asia and Australia. With inflammation networks, this grew further. The diversity incentive programs, which were connected to the seed networks, grew further. And then importantly, in some of the recent calls, including pediatrics, this has become increasingly international. And this is really representative of, I think, 2 key things. One is the engagement and help from all of you. Again, coming to HCA Asia meetings in the past has really helped us identify and connect with this community. And so we really want to hear what you're interested in and your priorities. And also the opportunity for current groups and current grantees to form those connections and reach out and form collaborations with scientists that really have excellent tools and connections around the world. So I would encourage you to get involved and connect with the current CZI grantees, and then also keep your eyes out for upcoming RFAs and opportunities to be funded or to apply. Okay, so let me tell you very quickly about a few of our recent work. So we have, over the last 3 years, been funding a large network of groups referred to as the CZI seed networks for the Human Cell Atlas. And the progress of the seed networks has been notable, but with that progress has also come a number of different gaps within the underlying data. One of those gaps was in tissues and samples from those early in life, so from children, so after birth up to adulthood. And so recently we announced a new round of opportunities earlier this year, and the eventual grantees that we're referring to as the pediatric networks for the Human Cell Atlas, total of about $35 million in funding and 134 PIs. again, distributed pretty broadly, 15 different countries across a number of different organ systems. Wanted to highlight just one kind of nice example, again, on this thread of international collaboration, and this is an effort to generate an atlas of the nasal mucosa. And I think everyone's eyes have been opened to the power and the importance of the nasal mucosa over the last few years. So obviously an incredibly important tissue for infectious disease, as well as other barrier functions. And of course, something that is highly dependent, not just on the ancestry of the individuals, but the environment that they're living in. And so a great opportunity and a critical opportunity to have this work include collaborators from around the world, including India, Bangladesh, the Gambia, the US, and many others. So 7 different sites to start with. Now, another critical aspect of all of this is representation. And as Partha has already mentioned in his beautiful talk, historically genomics has not done a good job here. And the representation, ancestral representation here, is historically very poor and heavily stunted towards European donors. Now, we know that representative science is simply better science. There's no argument that would argue against this. And beyond that, also having teams that are inclusive and equitable. So not just the participants, but the scientific teams doing this work. And again, I think Partha did a wonderful job of framing this. So this is something that we feel very strongly about and are looking to support and help fill some of the gaps and certainly avoid some of the, these disparities that have existed. So by doing anything we can. And so this has resulted in another additional call, the ancestry networks. This was announced very recently. Just earlier today, all of the project teams as well as the projects are now available on our website. And so I'll, again, with the time allotted, I'm already out of time, would encourage you to go look at these if you can. One wonderful example, of course, is an expansion of one of the earlier seed network projects focused on the Asian Immune Atlas and work that involves Jay and Cham and others. Okay, so let me very quickly go through cell by gene. And apologies for being a little bit over here. So cell by gene is designed to have an easily explorable interface for large scale single cell biology. So it is only focused on the derived data set. So you'll hear more about the efforts within the data coordination platform for raw and standardized processing, but it's designed to make visualization of multimodal data sets very easy. It is currently hosting about 20 million cells worth of data, about 214 data sets. This number is growing very quickly. And these data sets are all open. They're available. They come with a standard schema. So the data is interoperable and it has, everything has consistent ontologies for the cell labels as well as the author's annotations. So it's a great source for digging deeper into these data sets. On the topic of digging deeper into data sets, I do want to highlight a current open funding opportunity. So this is for what we're calling the data insights. So using available data sets, the data has to be openly available, such as the data within Cell by Gene or within the data coordination platform, an opportunity to apply and to bring questions that you have or to develop new tools for getting more insight from those currently available data sets. So this will be open until December 14th. But I'll also highlight it's the first of 3 cycles. So if you, for some reason, don't make this one, there will be more opportunities. So please stay up to date and keep your eyes out for these kind of opportunities. With that, I will thank you. Apologies for going a little bit over time.
Thank you, Jonah. Shall we take the question now, or do you prefer to have it all discussed at the end of this session? Just go with it.
Maybe there's a one question from the audience.
One question maybe.
Maybe that's, I don't know if you could see the question from Irene. I don't know.
Yeah, there are a couple of questions from the chatting board.
So I see, oh, I'm sorry, go ahead.
Right, so from Irene, there's a question saying from Kristen Wells. I'll go with the Kristen Wells question first. Can basic research fulfill the promise of local benefit without a path to benefit? In which case, how important is addressing disease atlases to the equity effort?
Yeah, so I think this is probably a larger question than I'm prepared to answer, and Partha and others may want to chime in. So I think disease atlases, of course, are critical for understanding the kind of the path to equity. I would say, and I think we saw this in COVID, that many diseases, including those locally, can be at least clarified by a representative reference atlas, right? It may not be the total question. I think there's a good argument to be made that part of defining normal is to understand disease and how the systems change and adapt. The immune system, of course, a beautiful example. So I think it's important, but I don't know that it is absolutely critical. I think there's a lot to learn and a lot to be gained, even by these first steps in the focus. I think representation is critical, however, and that must be achieved for.
Right. I'm sorry for giving you the distraction. Anyway, so I'll just go for one more. I mean, just to seek for what you think. So there was a question from Irene saying that so providing training grants for young researchers from countries where infrastructure just cannot meet single cell demands. What do you think of this, like international training grants or young researchers? Would that help the countries to promote the single cell genomics research?
Yeah, it's a great question. So to be honest, this is not a type of grant that we have offered in the past, with the caveat of the definition of infrastructure here. I may not be entirely sure on. So, for example, for data analysis, perhaps Irene and others might have access to some compute infrastructure that would make it possible to reanalyze or be involved in the analysis of data that they've also worked to collect the samples for, even in the absence of the generation of the data. I think there's always improvements for the infrastructure, but I know this is something where I would love to chat with Irene a little bit more. I wish there was a coffee break afterwards, because it's a great point and something that we should look towards.
Right, great. Thank you very much, Jonah. So I'll hand over my my mic to my next co-chair, Jay. I've been distracted a little bit, but anyway, Jay.
Okay. Yeah, thank you, Young-suk. Well, I think it's a great time to transition to our next speakers who would actually talk about the data coordination platform. As Jonah actually just mentioned, the data will be accessible and it is quite equitable and probably more infrastructure-wise can be more accessible than data production itself. So we look forward to hearing a talk. It'll be given by two speakers, Trevor, who is a computational genomic platform program manager at UC Santa Cruz Genomic Institute. And it will be co-speaker will be William Sullivan, who's a senior data wrangler and data engineer at the HCA data coordination platform at UCSC as well. So with that, we'll hand over the mic to Trevor and William, please.
Thank you, Jay. Let me just share my screen here. So today we're going to provide an overview and an update on the HCA's data coordination platform, what it is, what we do, and how you can contribute. So the DCP works with contributors to gather data on as many different human cell types as possible. This is a global effort requiring hundreds of individual labs. Researchers are profiling cells for their own research aims, and this results in a diverse organ and lab-specific data, and diverse collection techniques and data formats that suit the individual researcher's needs. The HCA community is multifaceted and has diverse data needs and challenges. Our broad community includes researchers in the lab performing experiments and isolating the cells and sequencing them, computational biologists identifying how to best approach and analyze sequencing data, and software engineers and tool developers creating new ways to work with the data. Each of these community members faces different challenges to accomplish their tasks. For example, an experimental researcher might need help contributing, analyzing, or visualizing data. experimental researcher might need help contributing, computational researcher might need help accessing data to use it for novel analyses. And engineers might need access to data to test it with developing tools that will better serve our community. All this diversity in data, tools, research aims, and community members, how do we bring everyone and everything together to form a meaningful atlas? So to do that, we adhere to FAIR principles to maximize the usefulness of the platform for every type of user. Platform must meet the needs of the researchers to develop the atlas. It must be comprehensive. There are many data types to describe human cells. It must be inclusive. We want to include all tissues and ensure all researchers can contribute to and access the HCA community. It must be organized. Data must be centralized and standardized. It must be accessible. Within the bounds of consent, data access should be simple. And dynamic. This is a living project that evolves with new data types, new techniques, and analysis methods. To leverage the breadth of consortia knowledge and data, we must ensure data is harmonized and described in a usable way with standardized metadata. Bring that data together in one space, a cloud repository. This will enable consistent management of data, enable access to raw data, metadata, and processed data. Processing raw data with consistent, accepted, and uniform pipelines will enable us to use data for the atlas. To make uniform processing and outputs possible, we need pipelines that scale to the size of the consortia's data. The pipelines need to be open and public for community review and usable by any researcher. Data must be regularly released with consistent processed data, accompanied with researcher-contributed analysis files. This allows data to be referenced for publication and collaborations. And data must be accessible and discoverable from a central location, a data portal, so researchers can use it. This is what we provide. So all these components make the data coordination platform a scalable platform where researchers can interrogate single-cell data. It's led by a team of engineers, bioinformaticians, and data wranglers. It leverages global standards and is proud to be part of the GA4GH community. It's more than a platform. It's a service where you can contribute data, tools, and ideas. And you can receive help with metadata and data submission to ensure your data is reproducible. Here we see how data flows through the system, from contribution to uniform pipeline analysis to user access with community tools. For data processing, we are focused on core processing of data applicable to a wide range of analyses. Currently, the DCP offers uniform processing with Optimus 10x and SmartSeq 2 pipelines. And you can try these pipelines in your own data in the Terra system. For accessing data, we provide a data browser. This allows users to find projects or build cross-project cohorts, export data locally or analyze in a cloud platform, or visualize data in external analysis portals. In this example here, we provide a multifaceted search of all the data in the DCP. And the image on the slides shows how we selected a species, Homo sapiens, an organ, pancreas, and an organ part, islet of Langerhans, and the resultant search shows 2 projects that we downloaded or exported and analyzed together. Data can be handed off to any tool supporting standard handoff mechanisms. Now, the data portal supports handoff with GA4GH standards such as the DRS, which is the data repository service of the GA4GH, and this handoff is to cloud-based analysis platforms. We also provide programmatic access to data in the DCP through a documented REST API. On the data portal, you'll find multiple third-party tools that we support. On this slide, we just show a few examples. These include cell by gene that Jonah mentioned earlier. We provide link outs for specific projects to visualizations in cell by gene, into the UCSC cell browser, the single cell portal, and others. And you can find a list of these data portals on the link that I've provided at the bottom of the slide here. Okay, so now we'll explore the data contribution process in more depth with my colleague, William Sullivan, who will continue this presentation. Thank you.
Awesome. Thanks, Trevor. I'm now going to kind of go back to the beginning of that data flow diagram, talk a little bit about data contribution. Yep, same one more. Yep, there we go. And so let's just kind of start the process of how data contribution works. First, you would go about researcher would contact the data wrangler team. We would then go about essentially formatting your data to DCP standards, which we will then submit to the DCP. There's a number of complicated processes that go with that, but essentially we will try to format it as best we can. And then at the very end, we will provide you with accessions for the data and metadata, and then that data will become publicly accessible. Next slide. There's a couple ways to reach out to us and the Wrangler team. The first is we hold Wrangler office hours twice a month, with the next one being this coming Wednesday, November 17th at 11 p.m. UTC. You can also reach out to us at the Human Cell Atlas Slack, specifically the Contribute Data channel, or you can email us at wrangler-team@data.humancellatlas.org, and you can reach out to us about contributing data or any questions you might have about the DCP. Next slide. Now I'm going to go into some recent updates that have happened since the last HCA Asia meeting. Currently, the DCP is sitting around 180 data sets available. As Trevor mentioned earlier, we've moved to a monthly data release cadence so we can give you guys more standard release times and do more snapshots more frequently for data. We've also started to allow contributor-generated matrices and analysis files. We're hoping that this will allow researchers to share published analysis. We also may be able to include batch-corrected and normalized results before standard methods exist for those. We're also hoping this will add power to analyses when juxtaposed with our uniform processing pipelines. And as Trevor mentioned, we've also made it a little bit easier to download and hand off to Terra all project-related data files, metadata, and matrices. Next slide. Here we show a graph of kind of how we've progressed over the last year for raw and processed data. With us, as I said earlier, 180 data sets and sitting about a little more than 20 million cells with the most recent update. Next slide. Now I'm just going to what our next big plan is we're trying to work with managed access data sets. We've been talking with the community and we realized that you guys are generating a lot of data that's requiring consent, whether that be patients with diseases, minors, or other protected groups. And we know there's a lot of questions that circle around managed access data sets, such as data regionality requirements, open versus controlled metadata, security compliance standards, as well as privacy standards. Next slide. To give you a little bit better idea of what we mean by open access versus managed access, when we define open access, we're talking about access to data without saying who we are, how we're using the data. Some examples of resources to do this are ENCODE and the 1,000 Genomes project. By managed or controlled access, we mean need to apply to gain access, generally based on patient consents or legal constraints. And these often include human research with genetic or phenotypic data. And some examples of that are the Wellcome Trust Case Control Consortium, the UK Biobank, or a subset of the GTEx information. Next slide. We currently are enabling some research to access managed data via the DCP. We do this by hosting the open access expression matrices and then providing an external link on the data portal to the primary data. And here's an example of that using linking out to EGA accessions on a project details page. Next slide. As you can see, we started initializing that in June of this year, and we've slowly increased, as seen in the green, with more and more of those managed access. Next slide. Oh, next, yep. Oh, back one more. There we go. But we realized that we'd also like to hold the primary data and the controlled access data. So we're starting to work towards building a data access committee with the idea of possibly adopting the data use ontology standards and with the hope of incorporating those into the DCP. Next slide. And this leads us coming to our upcoming data pilot, which will focus on managed access with hopes to get as much information about all the questions that I brought up previously, as well as talking about currently thinking about different pediatric research and consent-tied disease research and things like that. And with this, we're really interested in studies from HCA Asia members So we're really encouraging people to reach out to the Wrangler team at the email below, and then prefix your emails with HCA Asia. Next slide. Lastly, I just want to say we're trying to build this together. So we really love feedback on the data coordination platform, whether that's positive or possibly negative, but we take all of it relating to submitting your data, discovering the data, or using and analyzing the data. Feel free to send that feedback or any questions you have to either data-help@humancellus.org or the Wrangler team email. Next. Finally, I'd like to thank the Chan Zuckerberg Initiative for funding us, as well as the different organizations that are part of the DCP, such as the Broad Institute, UCSC Genomics Institute, and EMBL European Bioinformatics Institute. Next slide. I'd also like to thank all the contributors who work on the DCP. It's a massive project, as many of you can assume, and it's really been grateful to work with a lot of people that are all part of this, including Trevor. And next slide. And lastly, I'd like to thank the partnership across programs and funders. And yeah, so show them up. And then I'll go to the last slide, which will be the end of the presentation, and I can take any questions that you have.
Great. Thank you very much, Trevor and William. Excellent talk and right on time. Maybe a couple questions before we move on to our next speaker, actually. So you mentioned that in DCP, there has been a pipeline built in to process 3' and SmartSeq. So what's your plan to expand the pipeline for different technology modalities? And do you also consider spatial technologies and others to be part of DCP?
So I guess I can kind of field this one. I know at least in terms of like next pipelines, what we're planning on is starting to work through imaging and what are the major assays that we'd like to focus on for that, as well as standard processing pipelines. I know we have an analysis working group that's helping to tailor what direction we go for different projects and different analyses and assays. So I at least know imaging is the next one coming down the pipeline. And I think we are planning on trying to expand our Optimus pipeline to expand to not just into 3', but also 5'. I know there's work being currently done on that.
Great. I got your joke. Yeah, it's pretty.
That was not. Yes, it was intentional.
That's great. So looking forward to that. And maybe I guess maybe another question related to DCP is that you mentioned there are different third portal, like a visualization portal that gets plugged into cell by genes or other ways to visualize DCP data, which I think is going to be really powerful. But I think one of the key challenges that we face and see here in these meetings is data integration of batch correction or batch effects. So if you are dealing with multiple data sets that are in DCP and try to pull them out for visualization in cell by gene, for example, do you provide tools that will help to harmonize the data, or is this mediated by the user on the other end, or how does that process actually work out?
I think currently it would be mediated by the user on the other end, but I think our hope is we work pretty closely with the team at Cell by Gene. So if we can find ways to ease that process and take a little bit of the burden off the contributors or the users of the data, we would be all for it. I think the biggest thing is to be able to reach out to us and give us ideas of kind of what you're working with and what you're trying to do. And then we can try to put you in contact with people at Cell by Gene or with us to try to ease that burden a little bit more and to try to maybe incorporate things as we see them come.
Great, thank you. I'm asking a question on behalf of Shyam, who, as part of IDA, actually we submitted and managed access data to the DCP. Just wondering when is expected to be available for managed access data?
So I know currently we are working towards building managed access. It's, as I'm sure many people know, a very complicated issue to figure out how we mitigate that. As I said, we're currently kind of working towards a pilot with it. We do have the ability to submit expression matrices and then link out if they do have a controlled access repository that the data will live in. We're hoping, I think, it'll be the next major development for the DCP. And correct me if I'm wrong on that, Trevor.
No, I think you're right there. We can host the open access parts of projects at the moment and reference out to other repositories that hold the primary data, that's the actual managed access data. And in the future, next year, we'll be able to actually host all of those files within the DCP itself. But that is a more complex process.
Sorry, maybe related to that, Alistair asks that in managed access, who is the entity that holds it? Meaning, who's the owner of that data? Is it the HCA or Sanger or CCI in case of managed access?
I believe the plan is that the HCA will have its own data access committee that would approve and manage the data within the DCP.
Okay, I think there are other questions, but for the interest of time, we will move on to our next speaker. So thank you once again to William and Trevor for your very insightful talk on DCP, and we look forward to working with you more in the future.
Awesome. Thank
Great.
you.
Thank you.
Hyunsu, take it away. Thank you.
Okay, thank you, Trevor and William. So our next speaker is Varodom Charoensawan from Mahidol University. He will be talking on establishing and expanding single-cell biology network in Thailand. Without further ado, Varodom.
Okay, thanks so much, Hyunsu, for your kind introduction. So let me try to share my slide. Okay, can you all see my slide now? Right, so I just want to quickly second Jay's on the optimized farm joke, and that was proven. So even though I'm pretty sure William did that on purpose. All right, so now getting back on my own talk. So so today we'd like to share with you a little bit about how we have established and trying to expand the single cell biology network in Thailand. And we started doing this a few years ago, and back then there were very few people who were interested in single cell at all. So there's quite a lot of challenge to setting up this. So first, I'd like to give you a quick overview of how Thailand as a country contributing to the Human Cell Atlas. So right now, there are 6 members from 3 different research institutes who have already registered to HCAs. So as you can see here that now we're still quite small and close communities. So I guess you can think of Thailand as a case study where we start this single cell. Virtually nobody else was interested in this a few years ago. So about our own group, so from the beginning, all the effort has been done through close collaboration between my group, who are systems biologists, and Ponpan Matankasombat's group, who are immunologists. So in Thailand, possibly in similar to many other countries in Asia, the research labs are pretty small, and most of our lab members are grad students and intern students. So we have to combine resources, efforts, and also expertise from the 2 labs together in order to make this happen. And only last year that we managed to hire this 4 research assistants who work full-time on single cell study, especially the one that related to Human Cell Atlas. So I'd like to take this opportunity to quickly thank Sarah Teichmann and her team as well for all the help that helped us establishing this facility at Mahidol University in Thailand a few years ago. So 2018, that was when we were fortunate to get the first grant called Newton Fund from the UK to establish this facility basically. And once we get that the international grant, then we got some matching fund from the local grant agencies like TSRI and also from Mahidol University to get some machines. So we managed to get the first 10x controllers a few years back, the first in the country actually. And also at the same time, we got this Zephyr, which is the pipetting liquid handling robot, so that we can do protocol like SmartSeq2 as well. And in the next few months, we would obtain this BD Rhapsody as well. So apart from doing research on single cell ourselves, our group are very committed to expanding and disseminating our know-how to other groups in Thailand and those who are interested in. So one of the activities that we have done so far is to have this single cell genomic workshop a few years ago, and that was completely free. So this was 2 days activities when the first day we have talks from speakers from the UK and also from Thailand. And on the second day, we have the practicals, hand-on workshops. And it was attended by hundreds of people in Thailand. So we thought that was quite a bit of success because we managed to expand collaborations to quite a few groups in Thailand. So at Mahidol University, where Partha and myself are working, we already have two faculty members, new faculty members, Dr. Ananya and Dr. Varodom, who will also be speaking in this conference this afternoon, who join us. And also we managed to get some clinicians to become interested in single cell, and now they try to use our facility for to study the tumor heterogeneities and stem cell and so on. And we also in close contact with some colleagues in different institutes who also use single cell for different models as well. So in Thailand, it's still pretty close and small communities. And only last year, we were very lucky to become part of Asian Immune Diversity Atlas, or IDAS, which Shyam talked about yesterday. So last year, we together with Partha from India, Partha and myself, we joined IDA, and which are being led by Shyam from Singapore, Woong Yang from South Korea, and Jay from Japan. So as you know, that IDA project is currently being funded by the Charoensawan Institute or CCI. But in Thailand, we managed to get international funding as a leverage to apply for matching fund from our own local grant agency called PMUC. So hopefully with this matching fund, we would be able to contribute to even higher numbers of samples that we can contribute to the community and also to HCA. So I'd like to finish my talks by focusing a little bit on what we plan to do with IDA and with HCAs, particularly in Thailand. So if you look at Thailand as a country that situated right in the middle of Southeast Asia, you can see that we have neighboring countries surrounding us like Myanmar, Laos, Cambodia, and Malaysia. So interestingly, people who live in different parts of Thailand, they are historically, culturally, and also genetically different. And some of them have some relationship to people living in other countries around us, to be honest. So we thought that instead of just getting donors for the IDA from Bangkok, where Partha and myself are working right now, it would be even better if we can actually get the donor from different region as well because of their genetic diversity. And by doing this, we are collaborating with our colleagues in different medical schools in different parts of Thailand. So by doing this, we believe that not only that we can get diverse samples, but we can also closely collaborating with them and pass on our knowledge and know-how to different groups as well. So with that, I'd like to quickly wrap up. So in Thailand, we started doing single cell around a few years ago at Mahidol University. And we faced a lot of challenges, mainly because people believe that it would be something really difficult, and it's difficult to do it in Thailand. And by doing that, we were lucky enough to get some international fundings, namely Newton Fund from the UK and CCI from the US. And it's kind of like help us solve the first problem that, to convince people that we can actually do it. And then, by getting international grant, it's allowed us to get the local grant as well. So we thought it would work the other way around, but yeah, in the end, it seemed to work well. So right now, from our survey, the technologies and also awareness still seem to concentrate pretty much in Bangkok, which is the capital cities. So the question obviously is try to improve the awareness and not just to researchers but also to the public, and try to disseminate our competency to our collaborators and so on. And in the end, we like to try to encourage more Thai scientists to join HCA. Thanks, and I'll stop here and I'd like to take any questions if there are any.
Okay, thank you, Varodom. It was interesting, great talk. So I'll give you one question. So I'd like to, we'd be interested to know, so what is the policy in Thailand that the data produced in Thailand, can it be shared among the collaborators, or can the data be managed in a way that some people can get access to it if it cannot leave the country? How is it working in Thailand?
They're very good questions, but not straightforward at all. So I guess, as someone mentioned, it's different from countries to countries, but actually it's probably even different from one institute to the other as well. So I cannot answer this question for all over Thailand. But luckily, I mean, in Thailand, we have a sort of like national genomics program called Genomic Thailand. So in Thailand, we try to tap into that, and basically, if we can work, within that big umbrella. So we thought that it would solve the problem a little bit. However, as you can see, that we plan to collaborate with different institutes. So right now, what we're trying to do and spend a lot of time on it is try to work out and try to tailor-made policies and also IRB and so on to each one of them. So maybe to cut long story short, I think there's no easy way to do this, and you just have to put a lot of effort into satisfying each different institute.
Okay, thank you, Varodom. I think this issue and all the others could be discussed again in whole at the last of this meeting. Yeah, thank you, Varodom. So I'll hand over the mic to my co-chair, Jay again. Jay.
Great. Thank you, Hyunsuk, and thank you, Varodom, for your excellent presentation. Next, I would like to introduce Nam Phi-Vo, who's the director of the Center for Biomedical Informatics at the Doing Group Big Data Institute in Vietnam. So we look forward to hearing your talk. Go ahead, Nam.
Yeah, thank you. Okay, let me share my slide. Yeah, can you see my slide?
Yes, you can. Please go ahead.
Okay. Yeah, hi, everyone. So I am Nam Phi-Vo. I am director of the Center for Biomedical Informatics in Big Data in Vietnam. So my talk today is about sequencing Vietnamese genomes at scale towards precision medicine in Vietnam. So the main question of my talk today is why are we sequencing Vietnamese genome? So as you know, here there is a very serious bias between European population and the other in terms of number of individuals in the one study. So you can see population of in the right side, population of European is smaller than East Asian and South Asian and similar to African. That's most of the number of individuals in the one study is from European. And this trend doesn't change much through time in the bottom figure. Okay, so that's why we aim to sequencing more Vietnamese genome to reduce this bias. Okay. So here is our project named 1000 Vietnamese Genome Project. So we have here the whole genome sequencing of 1000 Vietnamese genome from diverse regions in Vietnam, North, Central, and South. And here we have the access from in October this year. So there are lots of users from around the world access here. So that one maybe is promising project. Okay, so based on this project, we are building the Vietnamese specific genome reference. So here is the first comprehensive reference in Vietnam, so that we customize the GICS38 reference with 1000 Vietnamese genome variants from 1000 samples with Illumina short-read sequencing. But we also use the ParkBio smart sequencing with 50 samples to do de novo assembly and structural variants. And here we build a graph-based genome reference. It's kind of new approach for genome reference. That it's difficult to construct or manage, but it will facilitate dealing with complex variants. It's better. So here we expect that it will improve the analysis of Vietnamese genomic data. So here is an example from a sequence map of the graph-based genome reference. We are building Vietnamese specific SNP chips. So here we use data from 1000 Vietnamese genome projects and the well-known disease-associated variants and also our partner clinical data to build a small chip, but with the same or even higher accuracy of the imputation and also biological functional SNP for Vietnamese. So we are also building the predictive model for common disease in Vietnam, and then we expect that it will be affordable for genotyping millions of people in Vietnam. So from 1,000 Vietnamese to million Vietnamese that will be sequenced or genotyping data. So we are also working on the disease risk prediction projects for Vietnamese, which we are sequencing and genotyping more than 2,500 samples. So here we have to detect Vietnamese specific disease associated variants. We are focusing on around 10 common disease from cancer to cardiovascular to diabetes, Alzheimer, Parkinson, and so on. So based on that, we are developing SNP chips and PIS model, polygenic risk score model for disease risk screening for Vietnam. So our approach is trans-ethnic genome analysis. So here is a collaborative project between Viet Big Data, Hanoi Medical University, and University of Queensland in Australia. Okay, so also another project that we based on this data as well is adverse drug reaction. So we are sequencing and genotyping more than 1,500 samples to detect Vietnamese specific ADA-associated variants. So we focus on SCAR, and that's severe cutaneous adverse reaction, and NSAID hypersensitivity, and DILI, drug-induced liver injury. And we are developing the predictive models for adverse drug reaction in Vietnamese patient cohort as well, using statistical and machine learning approach. So here we also have the regional research collaboration with CFORM, Southeast Asian Pharmacogenomics Research Network, and also Mahidol University in Thailand and Bioinformatics Institute in Singapore. So we aim to have broader impacts of PCX and ADR research in Vietnam, like revising national list of essential medicines of Vietnam, and also increasing ADR awareness of clinical practice in Vietnam. Okay, so here is a tentative plan for single cell sequencing. So we aim to get the Vietnamese population scale single cell RNA sequencing profiling. Here's a tentative project between Viet Big Data, Hanoi Medical University, and University of Queensland. So we have the time sample taken from 1,000 Vietnamese project and the other project at Viet Big Data as well. So we expect to be able to incorporate with other ethnic groups through HCA Asia community, such people today, and CFORM network and the other group. Okay, so here a little bit brief of our group here. So maybe Vietnam is a little bit new to HCA. So Viet Big Data is established by the group, the biggest private corporation in Vietnam, and led by Prof. Vũ Hà Văn, a professor of mathematics at Yale University, and we have around 200 members here with natural language processing, computer vision, medical imaging, and biomedical informatics, and the other organization, and a innovation foundation, and a training program. And our Center for Biomedical Informatics have around 40 members, more than 10 of them have a PhD degree, and I am director of the center. So I got PhD from the University of Memphis and did my postdoc at MD Anderson Cancer Center, University of Texas. And also I work as senior scientist from the University of Chicago before coming back to Vietnam to lead the team. So here's our collaborator. So we also have some well-known scientists in the scientific advisory board, like Pavel Pepner from UCSD, Ron Shamir from Israel, Robert Green from Harvard, and Ji Dang from London as well. Okay, so here's our network of organization and our project now here. Okay, thank you. So that's my talk today.
Great, thank you very much. Thank you very much. It was an excellent overview of your effort, and it's amazing how much enthusiasm and interest there are in genomic science in Vietnam, and we look forward to working with you in expanding the single cell genomics in your country. For the interest of time, maybe we'll move on to our next speaker, and we will ask further questions to other speakers towards the end of the panel discussion. Okay, so with that, I'll hand over the mic to Hansu. Go ahead.
Hi. So last speaker is Joshua Shou from Hong Kong. So he'll be talking on scalable algorithms for single-cell RNA-seq clustering, trajectory inference and cancer clonal inference. Without further ado, Joshua.
Hello, hi everyone. So hopefully you can hear me this time. I'm delighted to be here to share about some of our single cell analysis effort at Hong Kong. So I'm a bioinformatician. I spent many years in Australia and I returned to Hong Kong about 3 years ago. And I'm now at the School of Biomedical Sciences at the University of Hong Kong. I'm also a bioinformatics core lead at the Center for Panoramic Sciences, which is a center in which we do a lot of core genomic sequencing or mass specs or imaging services for the community. So we actually, in our center, we actually do a lot of single cell RNA-seq. So in fact, the first slide is not just about me, but it's really about some of the single cell efforts in my university. I can't speak for the whole of Hong Kong, but I can speak about my university. Actually, historically, we have had a lot of different labs, biological and medical laboratories who generate a lot of single cell data. And there are several that are particularly relevant to, because of the locality. Like in Hong Kong, we actually have a disproportionately high proportion of patients who have nasopharyngeal carcinoma or hepatocellular carcinoma, as this is a map of NPC, as you can see, is actually concentrated in China or Southeast Asia, and in particular within China, southern China, including Hong Kong, is actually one of those places with a disproportionately high burden of this cancer around the world. So we study a lot of NPC, HCC, and also other kind of developmental model looking at cartilage, skeletal systems, and so on. So, and this is done in collaboration with a lot of colleagues in the university. In addition, in addition to generating data from different tissues or tumors, we are interested in developing technology. So one of my colleagues, who Kevin Chia, is excellent in developing this kind of high throughput profiling system using for biophysical properties of single cells using ultrafast optical microscopy. And I have been collaborating with a colleague in Shenzhen, in the Harbin Institute of Technology in Shenzhen, using this microfluidic-based system to measure the elasticity of single cell, basically having single cell passing through a constriction channel under a microscope in a microfluidic device. And by the way, the speed and also the shape, the deformation of the single cell, we can actually infer the elasticity of the cell. So that's actually, these are different ongoing technological development that is going on in our area. So in my own laboratory, so I do bioinformatics, so we have been developing a lot of bioinformatics methods. So I'm comfortably share with you a few of these methods that we have developed so far, and hopefully this will be, and most of this we put them as open source software, so it will benefit anyone who are interested in this type of analysis. The first project that I'll talk about is using mitochondrial DNA mutations as endogenous lineage tracing markers. And the idea here is that, well, if you have human clinical samples, it would be a little bit difficult to do lineage tracing, which is to identify the actual genetic lineage relationship between single cells, as opposed to in a cell line system or a animal model where you can insert some kind of genetic marker into the cells and do proper lineage tracing. If you're a clinical sample, basically you can't do that. So the next best thing we can think of is to use endogenous mutations that are already in the cells. So in particular, we and also there are other people in the world that have been trying to use a somatic mutation in mitochondria, in the mitochondrial genome, as a way to, as endogenous genetic marker. It is, mitochondria is useful because they're small genome, generally accepted to have high mutation rate and have multiple copies per cell. So basically it just have high coverage, so it's actually easier to identify them. So we have been using this basic concept and develop a computational tool In fact, we have a pipeline, but in particular, within that pipeline, we think one of the hardest part is to identify, to do variant calling from the mitochondria. So we developed this tool called mQuad, and this is our bioarchive preprint, and you can find our tool here. And the idea here is we need to be able to identify reliable, good quality variant in the mitochondrial DNA that we can use for subsequent clonal identification. So we've developed this tool to do exactly that. This tool, mQuad, will sit in the middle of a pipeline that involves some kind of a variant extraction. So this is using a tool called CellSNP. And also after mQuad, after we identify a set of informative mitochondrial variant, we can use another tool called VirioSNP to identify clones. And we think the major application here is looking at cancer clones. In the center of mQuad is essentially the application of this very simple binomial mixture model that really look at, basically we want to have variants that have generally high coverage and also have variants that are not at, where the allele frequency is not at extreme, not completely almost 1 or not completely almost 0. We want somewhere in between. So that's basically why we basically compare 2-component model versus 1-component model. And that's basically how we favor SNPs that are kind of having a 0.5 or middle ground kind of a allele frequency. And we think those would be informative variants. We've applied it to a range of data. We applied to some SmartSeq data. SmartSeq is great because they generally have higher sequencing coverage per cell. So this is also true in the mitochondria. They have pretty good coverage. So we could quite reliably, using this public data set, to identify the different cell lines, and the different cell line does come from different origins. So therefore they could represent clones. And our method does perform quite well compared to existing method, which is called MGATK. So we think it's quite useful. We have another applied to not single-cell RNA-seq, but single-cell DNA-seq, and this is using a 10x platform. So in fact, this is the case where we can, compare the single-cell DNA sequencing data and also our mitochondrial focus variant calling clone discovery. So in fact, this generally works quite well. The other data set we've tried is a 3' enriched 10x single-cell RNA-seq data set. This type of data are very tricky because in general, you don't get good coverage at the mitochondria. What I should say is that we don't get even coverage at the mitochondria. So you only get good coverage at very distinctive location. So but even in that case, if your variant is located at the 3' end of a transcript, you will get it, and we can actually identify clones based on that appears to be biologically sensible. So that's the kind of computational methods that we have been developing. I'll just go through very quickly some of the other work. I know time is limited, but so this is another tool that we're developing. Again, this is the bioarchive tool. It is an R package for doing statistical differential composition analysis. In short, we think the key is to identify whether there is likely misassignment between cell type and that incorporate that misassignment model into our statistical model. And the question really is whether given two condition, wild type versus control, condition 1 versus control, condition 2, treatment versus no treatment, whether a cell population increase in size or decrease in size, whether there's significant difference in composition. And we think the key is to incorporate the differences, possible cell type misassignment during clustering. And basically by doing that, our method is called DCATS. It appears to avoid false positive, and appears to generally work well across a range of data sets. Sorry, I don't have too much time here. This is yet another tool that we've developed when we are doing clustering of very large data set. This is called FlowGrid. It's published in Bioinformatics, and this is the paper and the GitHub page. And the idea here is that this is a plugin that uses a modified version of DBScan, a spatial clustering algorithm, and it would, and we've implemented as a Python package that will work well with ScanPy. And yeah, this is the paper with the details comparison. It works well and works quickly. The last part is about visualization. So again, for people who generate very, very large data, exploring data could be interesting. So again, this is the GitHub page. This is our bioRxiv preprint. And if you have a few seconds, I'll just play this video. So this is the a quick demo of how we look at spatial. This is spatial transcriptomic data, and these are just two samples. And we can and overlay that with our in the single cell. This become a t-SNE representation of the single cell data. So it is a way to help us explore the transition from single cell type to the spatial transcriptomic data. So at the end, I just want to thank you, and again apologize for the technical difficulty in the beginning. At the University of Hong Kong, at the medical school, we are hiring. We are hiring bioinformatics, particular bioinformatics experts of any kind of expertise, so especially bioinformatics. At the faculty level, postdoc bioinformaticians, research assistants, students, and also just a side advertisement, in around next year, we are trying to run a workshop in Hong Kong on single cell data science. Tentatively would be June 2022. So yeah, if you have any interest and you happen to be in Hong Kong around that time, email me, and this is my contact information. Thank you very much.
Thank you very much, Joshua. Excellent stuff. I mean, it's very exciting, and I could totally see your contribution to various research groups here at HCA Asia, and even perhaps to DCP. Your last visualization on BZM and t-SNE was pretty cool. So hopefully it can be widely used. So is there any questions from the group? So I think for the interest of time, we will have to close this session. I just want to thank you once again, Joshua,